To date, biologists have cataloged between 1.7 and 1.8 million distinct species on Earth, and based on ecological models, that number represents only a fraction of the actual biodiversity existing in the biosphere today. Making sense of a volume that massive requires a rigorous analytical framework. Early humans categorized organisms by utility, identifying which plants yielded food, which fibers provided clothing, or which timber offered shelter. Aristotle developed the first formal scientific classification system. He applied a binary logic to the animal kingdom, dividing it based on the presence of red blood, classified as eneima, versus the absence of red blood, termed anema. He applied a similar morphological proxy to plants, grouping them by structural size into herbs, shrubs, and trees. Centuries later, Carl Linnaeus formalized this approach, publishing the two-kingdom system that divided all known life into plantae and animalia. The Linnaean model relied on a single structural proxy to determine an organism's kingdom, the presence or absence of a cell wall. Using highly visible traits to sort data is intuitive, but building an entire planetary classification system strictly on external morphology creates a brittle framework. It cannot support the true complexity of life. The invention of advanced optics revealed a hidden universe that defied simple visual categorization. Biologists were suddenly forced to place microscopic single-celled organisms like Chlamydomonas into the same category as massive multicellular organisms like Spirogyra. The model ignored metabolic contradictions, grouping fungi alongside plants simply because both possessed cell walls, despite fungi being heterotrophic and plants being autotrophic. Down at the molecular level, fungal cell walls are constructed from chitin, while plant cell walls are built from cellulose. The two-kingdom model also placed microscopic prokaryotic bacteria in the exact same taxonomic box as massive eukaryotic angiosperms. Organisms like Euglena broke the system's logic entirely, as they exhibited plant-like photosynthesis alongside animal-like motility and a lack of cell walls. By the mid-20th century, biological reality had outgrown its categorization framework. Morphological classification fails to account for microscopic data. Taxonomy requires a shift from evaluating an organism's external shape to evaluating its internal architecture. In 1969, R. H. Whitaker proposed a five-kingdom model based on absolute biological principles. The first axis of Whitaker's framework is cellular architecture, which establishes the split between prokaryotes and eukaryotes. The second axis, somatic organization, divides organisms by physical complexity, separating unicellular life from multicellular networks. The third axis, metabolic modality, categorizes how organisms acquire energy. Whitaker factored in specific heterotrophic strategies, Holozoic ingestion, saprophytic decomposition, and parasitism. Kingdom Protista fits into this framework as the specific coordinate for unicellular eukaryotes. Fungi occupy the multicellular heterotrophic space, while plants map to the autotrophic coordinate. Whitaker's matrix provides a coordinate system that maps phylogenetic and evolutionary relationships. Let's isolate the domain of the unicellular prokaryotes, Kingdom Monera. Kingdom Monera pairs the simplest physical architecture on Earth with some of its most complex metabolic engines. These cells lack a defined nuclear envelope and possess zero membrane-bound organelles. Physically, they are restricted to four basic shapes: the spherical coccus, the rod-shaped bacillus, the comma-like vibrio, and the spiral spirillum. Despite these simple forms, they possess advanced survival hardware, such as the bacillus endospore, made of highly resistant calcium dipicolinate. While most are heterotrophic decomposers or pathogens, they exhibit nutritional modalities impossible for higher eukaryotes. Some utilize chemoautotrophy, generating energy by oxidizing raw inorganic substrates like nitrites, iron, or sulfur. They also possess the enzymatic machinery required to fracture the immensely strong triple covalent bonds of atmospheric N2 to perform nitrogen fixation. Without this prokaryotic ability, the global nitrogen cycle would stall, starving the planet of the raw materials needed for amino acids. Structural minimalism reduces the cellular resources required to maintain internal organelles, allowing those resources to be redirected toward diverse metabolic pathways. In 1990, Carl Woese's research shattered Kingdom Monera and established the modern three-domain system. Woese moved past morphology to sequence 16S ribosomal RNA. Because ribosomes are essential to all life, tracking mutations in 16S RNA provides a molecular clock for deep evolutionary time. The genetic sequences revealed that Kingdom Monera actually represented two completely distinct evolutionary lineages that diverged billions of years ago. The first domain is Eubacteria, or the true bacteria. Eubacteria are defined by a rigid cell wall constructed from peptidoglycan, or murin. Peptidoglycan forms a matrix where horizontal polysaccharide chains are physically cross-linked by short amino acid peptides. The second domain, Archaeobacteria, encompasses extremophiles like methanogens, halophiles, and thermoacidophiles. Archaeobacteria withstand boiling acid and crushing pressure because they lack peptidoglycan. They utilize pseudomurin, reinforced by strong ether linkages and branched lipid membranes, rather than the ester linkages found in true bacteria. At the microbial scale, survival is determined by the specific chemical bonds of the cell's hardware. When we pull back from microscopic hardware to view the biosphere in its entirety, we see the interconnected engine of life. Biological classification tracks the actual movement of energy and nutrients through the environment. Heterotrophic saprophytes, the bacterial and fungal decomposers, dismantle dead organic tissue into raw, inorganic elements. Chemosynthetic bacteria in the soil oxidize these raw elements, cycling them into bioavailable minerals. Autotrophic plants combine these minerals with solar energy to construct the complex organic matter that shapes the environment. This autotrophic foundation is the sole energy source sustaining the advanced organ system-driven animal kingdom. This microbial foundation directly impacts human survival through pathogenesis and the synthesis of critical medicines like antibiotics. Whitaker's 5 kingdoms and Woese's 3 domains map onto this global energy circuit. The breakthrough in taxonomy occurred when science began asking how an organism is built and how it powers itself, finally uncovering the operating system of the natural world.